for those interesting presentations. So the floor is yours if you want to make, yeah, yes, and then.
I have a question for Pietro. Regarding the model you mentioned in the beginning, how does the model incorporate, let's say, successful drugs from unsuccessful drugs? Meaning a drug, even though it's a rare population, that a patient would be on for years compared to one where they would only be taking it for months.
Yeah, no, the model is sophisticated enough to do just that. So as part of the input is the survival rates. And so, of course, if the survival rate is very high, a pharma company is bound to make even more money because it's going to sell the medicine for much longer. If somebody is instead, unfortunately, is going to have the drug for much shorter, so survival rate is low, then the pharma company will get less. So that is actually incorporated. It's one of those parameters called A in the model, but it takes that exactly into account. It's important.
Great.
Pietro, first of all, I would say that this is the best lung cancer lecture given by economists in my life. Probably the first one.
The first one.
So I like your personal drug development model. However, there's one aspect that we have not talked about, actually two aspects. One is that how would this model be able to address the toxicity issue? Because in a way is that the molecule development is mostly protein-based, but then when you come to the human environment, it's so complex, it's unlikely any AI model is going to give you a prediction of what toxicity is going to be like. And then we still end up having to do the toxicity on the clinical trial before any agency like FDA would approve it. So that's one aspect. And second is that right now, a lot of our endpoints is still survival. And survival, again, is so complex that it's very difficult to just using a molecular prediction model to say that what kind of survival we're going to get. So I just want to hear your thought. Is there any other way that we can address this two issues of toxicity and survival, such that we can actually cut costs in the development?
Of course, that's an important question. We've been discussing a lot with Marina. Of course, Marina is the expert here, but I would just say that the reason why we are thinking that to do our proof of concept through drug repurposing is exactly to tackle the toxicity problem that you're mentioning, so that we can use the Argonne supercomputer to search for compounds that already exist and already approved for billions, as Marina would tell you, of possible molecular alterations. The second part on the complexity, that I will leave it really to Marina, that I have less to say there.
Yes, so what we are doing now, so the state of the art is called the SMILES. So you can do a graphical representation of the molecules, and there is one part that is called Toxifor, for which you can potentially predict the toxicity. So the big problem and the big limitation that we have now is that because the visual representations are just planar representations, for example, we can't predict the toxicity of the antibodies because the antibodies are big molecules and they can change during the time according to the protein. So, but I think that in the next couple of years, we will go there also to predict the toxicity of the big molecules.
But there's also the genomic predisposition and the pharmacokinetic in the body.
Yeah, no, I agree. I agree. So what we are seeing, I think, will go in 10 years. So this is the vision of 10 years. But going back to Arcela's lecture, where with the agents you can incorporate the polymorphs, the SNPs of the patients, the toxicity of the patients, maybe, we will arrive there for which you can predict from a chemical structure a possible activity and also the toxicity. We are not there yet, but you see, every 2 months there is something new that is coming. So
have a question, just a curiosity for Pietro related to the, and also for Marina, I mean for both. When we want to develop, for example, an in silico validation and a new molecule, not in repurposing, how much more or less it can take to develop the drug for?
So the case that Pietro showed of the CDK20 was completely a new molecule. So, and was done almost 3 years ago. So from this, the structure to the new compound, we know that it is about, 30 days. The problem is the production. But for the production, there is all the world of the 3D printers that are creating substrates already existing, for which I think also the production maybe will be solved in the future.
Maybe one question. Oh, sorry.
Pietro, I was wondering, how about IP data protection of such a process? Is that feasible at all?
Well, the way we think about it is the same type of IP, intellectual property of ChatGPT, Google, OpenAI, etc. So the process will be, to some extent, algorithmic based. So the idea is very similar in terms of the ability of patenting that component. It is similar to some extent.
So let's say what I was having a bit more problem to understand is the validation process. So let's say you like the FDA to believe this is, to base on your process rather than on drugs to be approved. So how many cases you need to validate the process? And which type of targets? So let's say, if I were the FDA, just to challenge you, I could put you the case of a tumor suppressor gene, because so far we have been unable to really get a drug to a tumor suppressor. So I'm trying to say here, what do you need actually to validate the process?
Yeah, so I will let Marina answer at some point, but the idea is that the overall population for the randomized clinical trial for the PDDP is the entire population of lung cancer patients. If somebody arrives, you don't know what they have. It's going to do the genomic screening, and then you do the part of finding what alteration this person has, and then all the various steps that I described. And then you do this for all the patients who keep coming at the hospital, with the usual sample of 500 patients, you should be able to start seeing whether the PDDP works better compared to the current practice, in which you randomize the two groups. The second group comes, and if you find out has ALK, does ALK. If it has ROS1, you do ROS1, etc. But the PDDP will be compared to the standard of care. You don't need all the many patients now that you have everybody at your disposal, including people who have very rare diseases. But again, that's really a medical question. So.
He answered well.
Well, one of the major concerns is the access to the expensive drugs. If you move to the approval of method scenario, then that means that the patents rise and the IP is solved, that is generalized, and that could facilitate the access in each country?
That is the hope. So now for the super rare diseases, we are talking about $500,000 and plus in cost per patient in the United States. Then, of course, outside the United States is about 1/4 of it. The hope of the new process is the reason why it's so expensive for something that is NTRK, which is a super rare, is exactly that you don't have many patients. The PDDP applies to everybody, and therefore goes back to lowering the price for everyone, democratizing medicine even for the most rare diseases. And that is what England is doing on something very specific, genetic alteration, nothing to do with our things, but we hope that we can get there. But the logic is exactly the same. It's not economical to generate drugs for individuals, but it's economical to have a process to generate the drugs for individuals.
But at the end, the price should
lower to a much more reasonable price now, about half of whatever it is. It's one of the numbers I showed, according to the model.
Thank you.
Okay. Thank you very much.
Well, thank you very much, and thank you to everyone to stay here yet at this hour. It's just as a reminder that we have dinner tonight in the Hotel Santa Catalina. It's going to be at 8:30. Okay, it's close. In the city, everything is close. It's near, but there is a transfer. I have to confirm, but there is a transfer from the hotel also. Okay, and tomorrow we will be here at 9:30 a.m. Okay, see you tomorrow. Or see you tonight.
Ya me voy a la cena porque llevo